Namaskar and welcome to CEC Gurukul Lectures. I am Dr. Kumar Shantanu, Associate Professor of Botany from Deshbandhu College. So, let us continue our discussion on various concepts of bryophyte biology. In our previous lecture, we have seen a glimpse of evolution of sporophytes amongst the bryophytes. One major aspect of the evolution of plants from aquatic habitat towards the terrestrial habitat was the evolution of conducting tissues. Bryophytes, being the amphibians of the plant world, they were the first one to colonize, to start inhabiting the terrestrial habitat, and these are the groups where the most primitive evidences of evolution of conducting tissues could also be understood. So, as far as the conducting tissues in bryophytes are concerned, let us talk about those tissues which could be called as the conducting tissues. The internal specialized conducting tissues, if present in bryophytes, they are restricted to gametophytic generation in liverworts, whereas they may occur in both generations in mosses. So, the bryophytes can be subcategorized into three groups: the liverworts, hornworts and mosses. Amongst the liverworts, we could identify some of these conducting tissues only in gametophytic generation, whereas in mosses we could identify in gametophytic as well as sporophytic generations. Conducting tissues are unknown in the case of hornworts or anthocerotes. Water conducting cells, or sometimes very popularly called as WCCs, with walls perforated by plasmodesma derived pores, they have been found occurring in calobryals and Palliviciniaceae. These are the members of Mesgarials and Tachycia amongst mosses. These imperforated WCCs, also known as hydroids, are present in bryoid mosses. A polarized cytoplasmic organization and a distinctive axial system of microtubules is present in the highly specialized food conducting cells of polytrichous mosses. Such food conducting cells are also termed as leptoids, and amongst the less specialized parenchyma cells of the leafy stem, they are also found, and they are also found in the seta of other mosses including sphagnum. So, as far as the first identification or first finding of such conducting tissues are concerned, we have found such traces in the form of WCCs or water conducting cells, also termed as hydroids, and the food conducting cells FCCs, also termed as leptoids, and we could find these traces of hydroids and leptoids in some of the bryoid mosses, in also parenchymatous cells of leafy stem, and also in the seta of sphagnum. So, we could very conclusively say that the evolution of conducting tissues have already begun from the group bryophytes. A similar organization suggested to reflect specialization in long distance symplastic transport of nutrients. This also occurs in other parts of the plants in mosses, including rhizoids and colonymata. They may be absorbed in thallus parenchyma cells of liverworts also. Perforate WCC in the calobrials, mesgariales and tachycia and hydroids in bryophyte mosses probably evolved independently. So, we do have traces of WCC and FCCs which have evolved probably independently. But because of the fundamental differences in the developmental design, homology of any of these WCCs or FCCs with tracheids is highly unlikely. And similarly, the FCCs of bryophytes, they present highly distinctive characteristics and they cannot be considered homologous with the sieve cells of tracheophytes. So, we do have some traces of these conducting tissues amongst various genera of bryophytes, but to say that these were the primordial tissues giving rise to tracheids or the phloem tissues is very inconclusive. So, let us try to understand the origin of vasculature amongst bryophytes and then amongst the pteridophytes. The emergence of tracheophyte based vascular system of land plants had major impacts on the evolution of terrestrial biology in general. It facilitated the development of plants with increased stature, photosynthetic output and ability to colonize a greatly expanded range of environmental habitats. In combination with prevailing global climate change, which included dramatic changes in atmospheric carbon dioxide levels, this gave rise to the development of the cuticle and stomata. These important adaptations reduced tissue dehydration and increased the capacity for exchange of carbon dioxide, thereby enhancing the rate of photosynthesis. Following acquisition of these two traits, early land plants evolved cells specialized for long distance transport of food and water. And irrespective of plant groups, these cells became arranged end to end in longitudinal files having a simplified cytoplasm and modified end walls designed to increase their intra and intercellular conductivities respectively. The two essential functions that are performed by the vascular system amongst archegoneti, they are the delivery of resources, that is water, essential minerals, sugars and amino acids to various plant organs, and they also, in addition to the delivery of these resources, provide mechanical support. The development of vascular tissue by land plant is generally recognized as one of the most important evolutionary steps in the colonization of terrestrial habitats. Both bryophytes and tracheophytes have developed conducting tissues consisting of cells specialized for the distribution of food and water throughout the plant body. In land plants, the degree of cellular modifications of transport cells increases from the bryophytes, pre-tracheophytes also termed as non-vascular plants, to the early tracheophytes, the vascular cryptogams, on through to seed plants. Studies based on fossil records and extant bryophytes have established that developmental programs evolved to form specialized water and nutrient conducting tissues. Based on the fossil record, early pre-tracheophyte land plants appear to have developed simple water conducting conduits having smooth walls with small pores, likely derived from the presence of plasmodesmata. We can see similar structures present, for example, in some of the mosses, the most ancient being termed as water conducting cells and the more advanced being the hydroids of the peristomate mosses. Here we can see in a cross section of a leaf of a moss that in the center we have the presence of hydroids surrounded by leptoids. The plant vasculature is a sophisticated system that has greatly contributed to the evolution of land plants over the past few 100 million years. The formation of vascular system is a well organized plant developmental process. The remarkable event of evolution from bryophytes to pteridophytes was the evolution of dominant and independent sporophyte phase, which symbolized the true roots, stem, leaves and vascular tissue. Present day plants with leaves and branches are the result of continuous evolution in the axial sporophytes of the primitive land plants. Earliest vascular land plants such as Rhynia, Horneophyton, Xylophyton, Stereozyton had a simple dichotomously branched axis and bore sporangia terminally or laterally. The developmental trends in the conducting tissues of mosses elucidate that bryophytic conducting system can contribute to defining the structural level of conducting tissues in the ancestral types of land plant that gave rise to bryophytes and tracheophytes. The conducting tissues of mosses begin with the phloem-like cells, the leptoids, and then the xylem-like cells, the hydroids. Leptoid is a cell type which is found in both phases of some mosses and resemble the primitive sieve elements of vascular cryptogams and particularly the ferns. Because of this similarity with vascular plant sieve elements, Benke suggested using the term sieve element of bryophytes to describe leptoids. Although the developmental process amongst these two, the sieve elements and leptoids, are completely different. As in the case of tracheophytes, the food and water conducting tissues of mosses are spatially related. In a transverse section of the gametophore, the food conducting system known as leptome is located as a sheath of cells which surrounds the central cylinder of water conducting cells. The leptome consists of the leptoids and associated parenchyma. Now, as far as hydroids are concerned, they are the water conducting cells. The term hydroid was first used by Pottoni to describe the tracheary elements of vascular plants and has been widely used to designate the water conducting cells of mosses. Hydroids are located centrally in the gametophytic axis and comprise what is known as the central strand. Collectively, the central strand of hydroids is termed as hydrome. You can see these diagram where there is a transverse section through the hydrome in the rhizome of Polytrichum commune, and similarly the TS through the leptoids of Polytrichum commune. Here we can see very clearly that these hydromes has a different cytoplasmic content as compared to the leptoid cells. In the leptoid cells we can see large vesicles which help in the transfer of nutrient resources contained in these vesicles. Now, studies of bryophytic conducting tissues in the last decade have revealed a number of similarities in the ultrastructure and physiology between leptoids and sieve elements and between hydroids and tracheids. And because of these similarities, the argument of parallel evolution of bryophytes from a non-vascularized ancestor is less tenable than the argument for the evolution of bryophyte from a vascularized ancestor. This unrevealed collection of similarities should be considered ancestral, just as a cuticle and stomata in mosses are considered persistent ancestral features. So now let us move on to next concept in the bryophyte biology, that is the dispersal of spores amongst various genera of bryophytes. Because during the course of evolution, as plants moved from aquatic habitat towards the terrestrial habitat, amongst various key evolutionary changes, the change in the mechanism or adaptive mechanisms for the better dispersal of spore was also very eminent. The sporophyte generation is very short-lived. It comprises of a capsule which produces spores by meiosis and a stalk which holds this aloft, the gametophyte generation. Now the spores, once released, are dispersed by air currents, and once they settle somewhere moist, they germinate. This recommences the gametophyte generation. The spore first produces a filamentous stage called as protonema, and these cells are full of chloroplasts. So it is very eminent that better the mechanism of spore dispersal, better will be the rate of survival. So amongst the liverworts, let us understand how the spores disperse. Under dry conditions, the liverwort capsule splits into valves or segments. Now this splitting exposes the spores and elaters. Because of the ambient atmospheric humidity, elaters undergo hygroscopic movements, and these hygroscopic movements, they cause spores to be flickered into the air. Such kind of dispersal mechanism can be observed in Palea, whereas amongst the mosses there are certain groups of mosses which do not follow, but most of the mosses, they follow this simple mechanism where all moss capsules, they have an operculum and peristome. The exception to this observation are graphite mosses. So the general dispersal mechanism takes place with the help of calyptra, operculum and peristomal teeth. Under dry conditions, the calyptra, which is the remnant of archegonium, it drops off. Calyptra initially protects the developing or the growing capsule. So when the capsule is mature, the ambient atmospheric conditions are dry, the calyptra gets shaded off and the capsule is absolutely exposed to the environment. Then the operculum is shed as a result of water loss by the annulus. Annulus are those specialized cells which are found in a circular rim-like arrangement over the anterior tip of capsule. So these annular cells, as a result of water loss because of the ambient low humidity, they rupture or they break down, and the operculum, which was initially attached to the capsule, through these annulus, it gets shed off. Now with the shedding of operculum, the peristome teeth is absolutely exposed towards the outer environment. The peristome teeth, they bent outwards. This movement of peristome teeth is also maintained or guided by the ambient humidity. Once the peristome teeth bend outwards, the spores fall out of the capsule and they are carried by air currents. In case of Funaria, the tips of the curved peristome teeth fuse in a central disc. When the conditions are wet, teeth elongate and slits between teeth disappears. But when there is a dry condition, teeth shrink and gaps develop between teeth, allowing spores to shift out. Whereas in case of Polytrichum, the central disc or epiphyrum is very large and the peristome teeth are tiny. This peristome does not respond to moisture. So what happens in case of Polytrichum? When the ambient atmosphere is wet, epiphyrum loose, teeth does not stretches and gaps between teeth disappear. During dry condition, the epiphyrum gets tightened up or becomes stout, teeth is stretched, gaps develop between the teeth. This allows spores to shift out. So different genus amongst mosses, they have a very unique way of or unique mechanism of spore dispersal. One such unique mechanism of spore dispersal we can encounter in sphagnum also. Sphagnum grows in temperate swamps. Dead plants provide peat moss also. So the capsule contains air and spores and they do not have any peristome. As the capsule dries, it shrinks. This shrinking compresses the air inside and the pressure goes as high as 5 atmosphere and the operculum is shot off and spore is also shot off along with the operculum, and this shooting of operculum actually makes a popping noise. So we can see that there are different spore dispersal mechanism which we can observe amongst various moss genera. So this process has also gradually evolved and which has allowed the mosses and bryophytes to survive in a much better way on a terrestrial habitat. So we shall continue our discussion of these bryophyte biology concepts in our further lecture. In the lectures to come, we will deliberate upon the alternation of generation, and then we shall move upon the mechanism of heterospory amongst pteridophytes and also the formation of telomes or the telome theory, and then we shall also see how the stellar evolution has taken place amongst the archegoniates. I hope I have been able to give you a broad idea of how bryophytes have evolved along with its various physiology and biology throughout the evolutionary process. Thank you so much for your patient listening.